My presentation today is about neuroimaging and neurodevelopmental conditions. Obviously, I'm not going to review all that there is to know about that. There are whole journals that are about that, so I'm not going to get into everything. So I want to give you a bit of a insight into how to read some journals, some of the methods, and for those, is there anybody here that does neuroimaging research? Yeah, so I'm sure you have way more in-depth knowledge about at least what your research is about than what I'm going to present today. So feel free to interrupt. I have no relevant disclosures. I get the money from all of the regular places people get money from. Well, not all of them. I'm still working on some of them, but just a comment on some of my disclosures. I'm going to probably, based on the preferences of the communities, mostly use identity first language, but it'll probably be a bit of a mix depending on the situation. In most cases, I'll use conditions rather than disorders, but with the recognition that most of the criteria for diagnosis use disorders. It will pop out at times, and often I'll use disability when there's an impairment or a difference compared to the population, but in some instances when there is a lesser performance compared to the population, I will use deficits, but I'm going to try to use traits more often because that's also what I think is kind of important, is that some of the features that we measure in kids with development, the traits are variable across the population, and that's what I'll be talking about. So just acknowledging that and some of my limitations in language. So neuroimaging includes a bunch of different techniques. I'm mostly going to be talking about MRI because it is one of the most common ones. It's one of the ones that has some of the best examples, and it's the one I know the best, but there are others, and I'll just mention them as well. One thing as a physician that I get asked a lot is, should I get an MRI for my kid with this condition? And there is lots of things in the news media, in social media. This is from Attitude magazine, which is a pretty well-respected ADHD-related magazine for families and professionals. And it has here, MRI scans unlock treatment and symptom prevention in ADHD, but which makes it sound like all your kids with ADHD should have an MRI. But even in the very first line on this article is no, they should not. Unequivocally, there is no indication for MRIs in neurodevelopmental conditions unless there's other reasons. But you can understand why families ask me about this all of the time, because it is in the news media and online all over the place. So why do MRI research in autism and ADHD when there is not, at least now, any clinical indication for doing that when it is a neurodevelopmental condition? Of course, kids with neurodevelopmental conditions also have other things that would prompt them to have an MRI. Higher risks of seizures, high risks of different intellectual disabilities, or an abnormal neurologic exam. In that case, they would get MRIs, but for those other indications. So there are lots of reasons to do MRI research if there's no indication right now that there's a clinical reason to do it, at least at this time. Some of the proposed reasons are or one that comes up a lot is to justify neurodevelopmental conditions as a biological process. I will talk a little bit about that, but I really feel like that's not why I do MRI research. There is proposals of using MRIs to, in the future, diagnose these conditions. That's also not why I do MRI research and why I don't think it's a great reason to do MRI research. We'll talk a little bit about that. Another one is to understand better the etiology or pathophysiology of these conditions. What are the brain differences associated with different traits or different difficulties that people have, and what may contribute to poor prognosis in different conditions. I feel like that's at least getting more to the answers that families and people with these conditions are looking for. And then understanding prognosis or response to treatment or interventions. That's what I'm really hoping, at least in the future, that some MRI research will help us with. But taking a step back, one thing that, and if any of you have me on your thesis defense committees meetings for neuroimaging, one question I love to ask everybody is about the neuroembryology. I'm a neurologist. I like that. I was taught by Harvey Sarnat, who is like, that's his big thing. But you have to understand how, if you're going to look at how the brain is constructed later in life, you have to understand how it might have gotten there. So one thing that is very important to put into context when you're imaging a neurodevelopmental process is the normal development of that process. There is a patterned order of events in neuroembryology that extends, as we know, well past birth into teenage years and 20s. And then if you want to take it further, I don't do adults, but there is that decline process on the other end of the lifespan. And if you don't put your research of neuroimaging and how the brain is functioning in that context, you won't be able to understand your data. And so here's some examples. So if I go back, you can see the processes of first the neurulation and then neuroproliferation, differentiation, right through to programmed cell death and the pruning of synapses and the myelination that happens to insulate all of those connections past birth. And so here's some examples of that. So this is Atlas of Normal Myelination, where you can see T1 and T2, which are two different MRI sequences that can highlight different tissues in the brain differently. T2, you can see in the white matter, starts at birth as kind of pale gray, and as the white matter gets myelinated, that fat's laid down, it gets darker up to 2 years, and then likewise on T1, the myelinated fat turning out white on T1, you can see it in the cortical spinal tracts, and then as you go through, now you have myelination through all of the connections in the cerebral hemispheres, with the frontal lobes being later to develop. On MRI, on a clinical MRI, it looks myelinated to a mature level at 2 years of age, but if you're looking at more subtle features that we can get on more advanced research scans, that process happens well into later years with again, you can see the trend to the frontal lobes being the latest to develop. And this is on fetal MRI. I love fetal MRI. So cool, seeing a brain being born. Through the ages of gestation, you can see that process of the gyrifications and sulcations forming. And so obviously if you're doing research on a term baby versus a 2-year-old kid, you're going to have to take that into consideration, what the differences are going to be. The other process is that functional development in the brain. So the connections that are made and how those connections are used. On MRI sequences, we use functional MRI for that. This, I believe, is actually from an EEG study, which showed some cool trends from neonates, newborns, through to adults, and this is using the default mode network, which I think I'll talk a little bit more about later. But as we develop those connections through our experience, through especially the preschool and into adolescent ages, the connections in the brain that underlie some of our functioning, they start very blobby and topographical. The connections are mostly local, and then as they get refined, as that process of pruning the extra connections happens through childhood, those longer range connections get stronger, they get more established, they get less plastic, and you can see that you have these blobby areas of the default mode network that now become more localized and specific in the adult. And that is also something that we can see on MRI in our research, but is the process that we know of through normal development. This is just one way that this has been studied, is with, and getting into some of the fun math that happens in neuroimaging. Using graph theory metrics, we can see this as increased clustering with longer, stronger connections. And graph theory is just one way to quantify that. We now have more established focal areas of, say, language. It's more established in certain parts of the brain, and those connections in the language network become stronger over a longer period of time, or a longer distance in the brain. Just like anything, and this will be a lot of the same points from the previous discussion, there are a lot of pitfalls to doing neuroimaging research, and getting to causation versus correlation. Lots of those things are fundamental to neuroimaging and being critical of neuroimaging research, just like any other type of research. But particular to neuroimaging, confounding factors that we know of in neuroimaging generally, but need to be especially considered and especially critical of when we're doing children, when we're doing neurodevelopmental research, are things like accounting for non-physiologic factors, motion. I do a lot of MRIs on kids with ADHD. We have to be very, very careful about motion. That is a confounding factor that we have good reason, and we have data to show kids with ADHD are going to move more in the MRI than those without ADHD. And so every time I look at my imaging data, I have to be very careful that I'm not just picking up that increased motion in the MRI and not actually a difference in the brain. Not accounting for misattributing differences to confounds. That extends from that, taking the data right through to the analysis as well. In pediatrics, their brains are different, right? So they're smaller, the myelination is different, and depending on what you're looking at and the analysis that you're using, if you're not accounting for that appropriately, you're going to be misattributing differences. Insufficient data collection. It is hard to recruit. It is hard to get decent data. If I am eliminating all of the kids in my study who move too much, now I'm going to have smaller and smaller sample size. Doing short, one of the scans is the functional MRI scan. I'll get into a little bit about that, but you need to take that over a period of time. If you're cutting out all of the period of time they're wiggling, you're going to have shorter and shorter, you're going to have less data. It can be very hard to get appropriate signal out of those scans. But also, just as in the epidemiological data, we can't measure everything in kids, right? So there are likely also missing data characterizing the population in a lot of studies. Most of the studies are cross-sectional, and we got a good introduction to why that isn't always great. There are longitudinal imaging studies, but MRIs are expensive, and so cross-sectional studies are the norm. And then problems with correlation or with interpretation. Correlation versus causation, and when you are doing a study to see if there's a brain association associated with the trait, that you can get into a very circular argument about what is associated with what, especially if you are not accounting for all the confounds, and if you come to that study with a particular idea of what you're going to get out of it. You'll probably find it if you're looking. This is just one example about incomplete data. So, and this is better now, but for the longest time, we know the cerebellum is involved in other processes than motor. I am a neurologist, I do a motor exam, I do particular motor techniques to see if there are cerebellar problems. We know the cerebellum does other things. It's involved in language, it's involved with emotional processing. But almost all of the studies, usually they're fMRI studies, that look at brain associations with things like language, don't include the cerebellum. So if you are going to go and look into the literature to see what associations, there are neuroimaging associations with those conditions, most of them won't include the cerebellum. We cut it off at acquisition, and even in processing, a lot of the pipelines just take the cerebellum out.
Because the cerebellum was one of the first part of the brain to be involved in autism with Kursha works in the 1980s.
Yep.
So why has it disappeared?
Yeah, it's a good question. Well, one thing is that another area of the brain that a lot of people really are interested in is the frontal lobe, and just how you align, and if you want to get highest resolution and you're aligning that box of where you're getting data, if you're making sure you get the frontal lobe, a lot of people will just end up sacrificing the cerebellum. It's just a choice that's made in the acquisition. Happening less and less because our MRIs are better now. So to get that resolution in a shorter period of time, because the elements to taking an MRI are resolution, how fine that picture is, how long you want the scan to be. The higher the resolution, bigger field of view is going to get longer, and then the field of view, how big that box is. So it's better because our techniques are better, but it was a choice, just a bias to the cortex. And then, like I said, in analysis, one of the reasons it's cut off in analysis, when they do the brain extraction, they extract everything but the cerebellum, is the cerebellum is different. It looks different than the cortex, and our analysis, whether it's segregating the gray matter from the white matter, those kind of things, they were built with that cortical bias. We need to make sure we're doing this really good for the cortex. Cerebellum being structured different, the techniques just weren't as good. Again, we're getting better at that, but that's just a very clear example of pitfalls. Another one of those confounding factors is that we don't account well in our analysis, or we are not always attributing appropriately our interpretation to confounding factors. We have done many studies where we know that there is a neuroimaging correlate of socioeconomic status. This is not surprising, right? When we look at the potential contributions to development in all kids, socioeconomic status is a big one. So it makes sense that that would then leave an imprint on how the brain is structured through development. But very many studies of neurodevelopmental conditions will look at those brain correlates of different traits like executive function and not consider or measure that socioeconomic status could be different between those groups as well. I believe, was it yesterday you guys got a talk from Evdokia, and if she gave a similar talk this year as she did last year, in the cluster analysis, they showed that a lot of the brain groups and differences between groups and brains can be attributed to socioeconomic status, which we know at a average or group level is different between families that are affected by neurodevelopmental conditions and those that aren't affected by neurodevelopmental conditions. So attributing a brain difference to executive function in an ADHD group, you might actually be just seeing the difference in economic and academic opportunities that's already different between those groups. And those things in imaging studies are not often measured. So one thing, if you're trying to take into account all these different factors, if you're trying to measure both the behavioral traits and some of the things like socioeconomic status, and trying to get more data to be able to have more solid analyses, we often use databases. And here's just some of the examples. Databases like the ABCD database is a prospective longitudinal population-based study. Like any population, there is about 10 to 15, or about 10%, 8%, if I remember right, of kids in that database have ADHD. Because of their criteria, there's less autism, but there are also retrospective databases like Abide, where they took a bunch of studies that were autism studies and combined them together. And so you can see there's already a lot of difference between those approaches and what you might be able to get out of those different studies depending on the numbers and the data that's available. But there's a lot of them, and it's handy to know a bit about them if you're looking at neuroimaging. This is an example of an analysis where they compared some different studies. So in the pink, we have the Enigma, which is a large consortium of researchers who have contributed data. There's a emphasis on genetic studies in that group. And then the ABCD data is in the two different blues, which again is a population prospective study in the States. You can see that in the Enigma study, these are different volumes in different cortical areas, comparing ADHD to non-ADHD kids, that there's a lot more differences that are found in the different brain structure areas in the Enigma study in these pink. So you can see all these here. Most areas are different. Versus in the ABCD data, they have a couple of select areas, and the two difference between the blues is how many things they accounted for in their model, basically. So this is just an example of, you have to be at least considerate of what this construction of those databases to be able to understand the differences. If you have a study that is directed with, we'll say, similar to the previous population, when it is a study where they have recruited kids with a condition and kids without a condition, you're going to find more differences than if it is a prospective, more population-based database, because, and we know we've done studies, and others have as well, people who volunteer for research studies to be in a control group are doing it because they want to contribute to science, and they tend to be that super healthy, economically advantaged. They have time and resources and an interest in science and the ability to volunteer. And so you're going to find more differences. Not to say that that's not a legitimate analysis to do, but you have to consider your findings in that case. When you're doing big databases, there's also some of the confounds of, back to those non-physiologic confounds, we know different sites will be different, and that could be everything from the MRI machine that was taking those pictures to specifically where their recruitment is being done, there are side effects. So there's a lot of work being done on how to harmonize data so that we can have these big multi-site studies. We can get lots of kids with this expensive MRI data and analyze them together. And that's everything from platforms to standardized methods. So CPIP is an emerging Canadian platform where they are developing across a number of sites recommendations for how to take your MRIs and processes in your protocols so that that data is now more comparable at the acquisition, but then going through other harmonized methods. Combat is one where you try to statistically account for those side effects so that the hopefully your finding is more based on the brain differences and the imaging outcomes that you're looking for, and not just the fact that there are a lot more artistic rich people in California versus another part of the States. So going a little bit into some of the MRI methods themselves, because MRI, and I always tell both my clinical patients and my research, be prepared, it's going to take longer than you think. We take lots of images in lots of different ways so that we can see things differently. So starting with structure, and I already introduced this a bit, the fundamentals are T1 versus T2. T1 is considered our structural scan. It can show very fine detail, with gray being gray and white being white, of the structure. And the T1's usually if you're looking at a cortical volume study, if you're looking at all the different volumetrics things, they would be from a T1, high resolution T1 scan. T2 is less common in research, but we use it clinically all the time because it shows areas of scarring, differences in the fat, differences in like water content. It shows those things up well. But you can see it's harder to differentiate the cortex from the white matter. But this is what we look for strokes or inflammation, that kind of thing. And then DTI is or diffusion tensor image, is a way of looking at the usually white matter, but the way that water moves in the scanner or in a part of the brain in the scanner. And I'll go into a little bit more detail about that. So some sort of hypotheses and things that have come out in neurodevelopmental conditions from structural scans. One that is at this point not discussed a lot is overgrowth hypothesis of autism. They noticed very early that in imaging studies that kids with autism on average had bigger brains, and more cortical volume than those kids without on average. And this is a study from 2011. And you can see that that is true, right? So the red is autism and blue is controls. On average, the reds got higher volume both in total tissue volume, white matter volume, and gray matter. On average is higher than the controls. But more studies have shown that actually the overlap, and if you're looking at the individuals here, there's a lot of overlap, and that's probably driven by some specific subgroups within autism. And generally, like in girls, it's less likely that this overgrowth hypothesis is generally true in autism. It's probably driven by a subgroup. And well, that's important information to know, especially if you're getting to doing more genetic imaging studies, which if I have time to, I'll get to, is probably that process of, again, going back to the process of brain development that might be related to. It is probably related to either increase in neural migration, but more likely a decrease in the synaptic pruning and the normal development of cutting down some of those networks through development is probably driving that brain difference in a subgroup of kids with autism. One of the hypotheses in ADHD is this delayed maturation, so that the frontal lobes particular are going through the same process of development, but at a later age, causing this delay in the development of things like attention and executive function, impulse control in the kids with ADHD. And so this is an example of this. They're mapping the cortical volume in kids with ADHD through ages 7 through 12. So purple is the typically developing, blue is the ADHD, is that you could see that refining of the cortex and the time to the peak thickness is much later in kids with ADHD compared to those without ADHD. And this is an example of looking at medication effects. So differences in typically developing kids, kids with ADHD on stimulant medication, and kids with ADHD that are not taking medication. And this is an example where the indication is that in those areas where there is likely a difference in volumes between typically developing and ADHD not on medications, is that those on stimulant medication, that might bring them to more like typically developing. That's interesting information, but the interpretation of that is very difficult, right? Is it the medication that is changing how the brain is developing, or is it the medication is now changing the experience that kids are having, say at school or within their peer relationships, that is driving that brain difference? And then we haven't gotten to all the methods. Maybe it's something completely different, right? Maybe the kids that are on ADHD medications versus those that aren't on ADHD medication, because this is not a randomized control trial, right? We didn't put one group on medication and one without. That choice of whether or not they're on medication, maybe that's a confounding factor. There's a lot of things to consider. So getting to diffusion-based methods, I won't get into this a lot because it can get quite technical, and even within DTI, there are a number of different processes. The newer ones are some of these high angularity, high resolution DTIs. And we did a MRI upgrade at the Children's Hospital about a year ago. They went from being able to do about 32 directions in 18 minutes to now they can do 60 directions at higher resolution in like half the time. With those new advances in the acquisition, there's also new advances in the processing and the things that you can learn from DTI. So I'm not going to get into it a ton, partly because I don't understand all of it, so, but it can get very technical. But the basics of it is that MRI measures water molecules, and diffusion tensor imaging within a voxel measures how free that water molecule is able to move in different directions. So this is a tensor, And you can see in this direction, it's able to move quite well, whereas it's more restricted to be able to move in these other directions. And that is an interesting way to look at white matter, because you have these axons and the white matter tracks that structurally change how the water molecules move. And we can do things like tractography and try to trace those tensors through the different voxels to see where those different tracks go. But you can also do things like how restricted in different directions the water is, to learn things about that white matter, how dense it is, how cohesive it is, those kind of things. And this is a study in typically developing kids that shows that in different parts of the white matter, it goes through that maturation process, starting at 5 into early adulthood. Again, it follows that process of those white matter changes becoming more mature into your 20s. I always in medical school, we got a lecture on brain differences in maturation, depending on and referencing a lot of the MRI things. And I remember very clearly the lecturer talking about how, so if you see a teenager smashing a bus shelter, just know there's still hope. Their brain is still developing. They're not done maturing, so don't write them off yet. I was like, yes, that's a good message to take from neuroimaging, is the teenagers, there's still hope. These are some of the things that we've seen in neurodevelopmental conditions from diffusion tensor imaging, where they first did the tractography through different major tracts in the brain, and then looked at the fractional anisotropy, how the fractional anisotropy is the difference between the most diffusive direction and the other less. The formulas are there. You can look at them more later if you like. And what they found was that in ADHD, there's decreased fractional anisotropy, which is interpreted a lot of the case as sort of less cohesive white matter compared to controls in a number of different areas. But on meta-analysis of this data, that was not significant in the pediatric group. So it only became significant, again, with that maturation process into adulthood. When it had reached the mature state, they could see those differences, but through childhood, they were not significant between the different groups. So then getting to functional imaging. Two basic types of functional imaging in MRI, or two different experimental setups. Task-based is dependent on knowing the timing of different things that are happening in the MRI. And it can be as simple, I like to use the very simple example of finger tapping. So if you want to determine which part of the brain is associated with finger tapping, you start your functional MRI scan. You take scans every usually 2 seconds or so. And you have a period of time where they're not finger tapping, so that's here. And then you get them to start finger tapping, and you compare this part, the on part of your task, with the off part of your task, and see where the difference is. And you can compare different parts of the brain. Here you can see the red line is an area of the brain that is associated with finger tapping, becoming more a higher bold signal during those periods of time. The other major type is resting state. So resting state is handy for pediatrics because you just put them in the scanner and tell them not to do anything. Unless you have a kid with ADHD who's really bad at not doing anything. So we often actually do our resting state with passive video viewing. So they watch a kind of engaging, but not, it doesn't have funny parts or emotional parts or particular aspects of a story. They watch this video, it keeps them still better, and you see how different parts of the brain, the bold signal varies. And so now this is not associated with anything task or anything that they're specifically doing in the scanner, but you take different parts of the brain and see on their time course if those different parts of the brain activate or have higher bold signal together versus more randomly. So here, the yellow and orange, you can see those two parts of the brain. When they change, they kind of change together, and so those would be positive functional correlation versus the teal colored, where if anything, it's the opposite, and that would be a negative or anti-correlation. And the reason to do those types of scans is you can then pull out networks that work together. So things like the default mode network, which I'll talk a little bit later, are associated with different traits or different functions, and you can study how connected or unconnected those networks are just at baseline in the brain. So this is an example of an fMRI study in autism and on meta-analysis of task-based studies. So again, they are doing things in the scanner and they're seeing which areas of the brain are associated with those tasks. They found that there was differences in amygdala activation, but they were not consistent in direction. So one thing, if you think of the amygdala, is associated with fear response, it's associated with emotional processing, other things as well. Some people theorize that that would be reduced in people with autism, and in fact it was for facial processing, but when they were doing different eye gaze studies, it was actually increased activation. So there is indicates that there is differences in the activity of the amygdala in the groups of people, at least with autism, but it's not that it doesn't work, it's that it works differently or with different stimulation compared to typically developing. And then, coming to the default mode network, one thing that's been found fairly consistently is that the default mode network has differences in its connectivity compared to people without autism. This gets to that kind of circular argument. In population or less clinical groups, they have associated the default mode network and its connectivity to things like introspection, theory of mind, and social cognition. But some of those associations come from people who are not great at things like introspection. So now we're attributing decreased introspection to people with autism because their default mode network is different when we got that from neuroimaging studies. So you do have to be careful of that sort of circular argument of what this means for autism as a condition. I think I'm going to skip over this for sake of time. This is an interesting example from ADHD. This is a meta-analysis of resting state MRI studies in ADHD, included almost 2,000 participants with and without ADHD, and combined them to see were there areas of the brain that were ADHD areas of the brain or networks that were associated with it. And when these are the spots here are all the different areas that were found both positively correlated and negatively correlated with ADHD. You can see that they're everywhere pretty much in the brain. But when they did the meta-analysis and combined them and then thresholded them appropriately for multiple comparisons, none of them were statistically significant. So that's kind of interesting. Means that there's not an ADHD network or an ADHD part of the brain. That kind of makes sense if you know all the things that could go wrong in different neurodevelopmental conditions. It makes sense that there wouldn't be just one area. And this is a study kind of comparative where they did a similar thing looking at resting state networks now in the ABCD database. So that very large 11,000 kids followed prospectively in the States. And they found, again, when they look at all of the different areas, there were areas that were more or less associated with ADHD, but you can see it's kind of everywhere in the brain, some areas more than others, and so this is actually the default mode network. You can see it's a little bit darker, but there were very small effects and they were widespread through the brain. So so that's pretty good comparative, using different techniques, using a different population and a different potential biases to show that a similar result. One thing that is also a theory, more of attention rather than associated with a condition, is the triple network theory. So again, the default mode network here, which is a task negative study. We know that it is most connected and most active when you are not doing anything particular, not engaged in a task, versus the frontal parietal attention network, which is most active when you are actively trying to pay attention to something and engaged in a task. That those during attention should be anti-correlated. So if you're engaged, default mode goes down, attention goes up. They should also regulate in the opposite direction. So if you're just trying to clear your mind and review your day so you can settle to go to sleep, it should go the other way. And that the theory is that the salience network, or also called the ventral attention network in some studies, helps you switch intentionally between those states so that you are paying attention to the things you should be paying attention to and not paying attention to the things like distractions and those kind of things. The salience network's meant to be the coordinator of those things. That's the theory anyways. That is not a specific theory to ADHD, but they have looked at that in ADHD, and there is some support that that coordination of networks, which wouldn't come out in the other studies, right? You can't just look at the networks and know if during different states they're anti-correlated or changing in their time variation as needed. You can't do that on a meta-analysis of a whole bunch of other studies. So this is a study where they looked at the variability over time and showed that the ADHD kids' variability of coordination between those networks was increased compared to typically developing controls. So it's not that their brain is not functioning, it's not that those networks aren't there, but that the regulation between those networks might not be as efficient in kids with ADHD, which I find interesting. Other MRI techniques, spectroscopy, and there are others as well that are meant to look at chemical or regulation parts of the brain. Spectroscopy, here's an example, just like a mass spectroscopy, is looking at or trying to tease out the relative amounts of different molecules in the brain. Usually that is done on because this requires a lot of data from a very specific part of the brain. It's usually based on a voxel. And so, but they are getting, again, with new techniques and better MRIs, they are getting new processes where they're starting to do whole brain spectroscopy and seeing, and then again comparing between different groups or based on different processes, if there are more or less, say, GABA or N-acetylcysteine, those kind of chemistry in different parts of the brain, and if that can teach us about some of the functioning. Other ones are things like arterial spin labeling, which can then see how the blood vessels and where the blood is going during an MRI scan, or MRI techniques, but other non-MRI techniques like FNIRS, based on, or measuring blood flow based on infrared, SPECT, PET, diffusion, CT, EEG, MEG. Most of these are either functional or chemical based, and I don't have time to get into any of them, but they a lot of them have limitations in pediatrics because of either tolerability or ethics, where you can't, PECT and SPECT have, you inject radio tracers. You can't really inject kids with radiation for a research study. They're not going to let you do that. You can do it clinically if you need to, but they're not going to let you do it just for interest's sake. One of the theories that has come out, and again has variable amounts of evidence, is this inhibition versus excitation balance in the brain, and that it's been proposed as a sort of common final pathway with all the different genetic contributions and other potential contributors that it's this imbalance in different parts of the brain might be underlying at least some of the behaviors in autism. And they have found lower GABA in the motor and auditory and frontal cortices, but it's been inconsistent. This is interesting though, because we do have medications that can help affect those different chemistries, right? We have GABAergic medications. So if it was found that it is decreased GABA, well, there's a lot of considerations, and that might point to either a specific medication choice for an individual or an ability to track how effective different medications are. So it's definitely not to the stage that it's actually clinically useful, but you can see how if we improve techniques that it might be. Again, I'm going to skip over this because we're running short on time, and I wanted to talk at least a little bit about sort of new and future directions. So if we get back to some of those proposed reasons for why are we doing neuroimaging research, like I said, to justify that it's a biological process, I don't know, we're biological entities, most things are a biological process, and whether it's a brain difference that's associated with a trait versus one condition versus another condition, honestly, I'm personally not that interested in that. I'm more interested in what it can teach us about the prognosis and potential risk factors overall, which in the imaging studies that I've reviewed is less important about one condition versus another. And I hope I've emphasized more having to do with that trajectory and the different processes that could be contributing to different behaviors rather than specific conditions. To diagnose or subgroup, there's a lot of research now about using machine learning to be able to diagnose autism versus not autism. One thing I haven't gotten into is that just like you can't inject kids with radiation for fun, you also cannot give them a general anesthetic to keep them asleep in your MRI study, which can be between half an hour and 60 minutes or even longer. So any neuroimaging study with groups of people with autism is not going to be representative of the whole population of people with autism. I would say in my clinical practice, maybe a quarter of them could withstand doing an MRI without general anesthetic. When we're doing it for a clinical purpose, we put them to sleep. You cannot do that ethically for a research study. So any imaging study of kids with autism is going to be a biased population or a biased sample of that population. And so if you're doing machine learning to diagnose, you're going to have data that's going into that machine learning algorithm that we need to acknowledge is biased, both from the autism sample side, but also, as we covered, the control sample side. And so while there is evidence that machine learning might be able to, in concert, neuroimaging in concert with other things, might be able to appropriately categorize people into having autism or not having autism with reasonable statistical accuracy, I'm not sure how useful that actually is, because the process of understanding a kid and their strengths and weaknesses during the diagnostic process, to me, is a lot more useful than just being able to put them into a category. So there's a lot of work in that area, and I think it's interesting, but personally as a clinician, not of any utility to me, even if they could get 100% accuracy. But that's my rant about that. Talk to me long enough, there's going to be a rant come out, just like always happens. And then other things like understanding the pathophysiology, understanding what may or may not contribute to an individual's presentation. We have to remember what we've already learned, right? We've already learned that things like socioeconomic status and economic opportunities changes kids' developmental trajectory. And so we can't forget about that when we're doing a study looking at neuroimaging. We need to include it in the context of what we've learned about neurodevelopmental conditions in general. And at this point, that translation to clinical is not there. Both the techniques are not good enough, the cost of the techniques are too high to justify it compared to what we're already doing. and then understanding prognosis or potential response to treatments. That's where I try to concentrate my research. But one thing to particularly keep in mind is that we want to make sure that just in all research in neurodevelopmental conditions that we are targeting outcomes that are interest to the people we're studying, right? So improving an outcome, if we can associate, say, brain imaging with, let's say, eye contact or something like that, that amygdala activation that changes whether it's facial processing versus eye tracking. Well, if that outcome is not of interest to the people that are affected, to the families that are affected, then why are we studying it? And that is something to always keep in mind in neurodevelopmental condition research, but is sometimes when we get into the weeds of all of the technical things of neuroimaging, we forget about. So this is an example of subtyping neurodevelopmental conditions. So using data-driven techniques. I'm not getting into a lot of machine learning stuff because I have a postdoc who's teaching me all about it, but is not my area of expertise. Is that in, and this is from the PON database out of Ontario, where they have kids with autism, ADHD, obsessive compulsive disorder, and a control group that they are following longitudinal, though this is a cross-sectional analysis. When they took both clinical features and neuroimaging features, they could subgroup them, but they found that a lot of the subgroups did not actually track along diagnostic groups. They were more associated with different clinical features and different behavioral features that had particular patterns in the brain, which I find very interesting because if I'm thinking of intervening on a behavior that a maladaptive behavior or area of concern, a cognitive concern or behavioral concern, knowing that it is not based on the diagnostic group, but more based on I'm better grouping those by clinical features is important to me when I am thinking about what kind of treatment or therapies might be most useful in that condition. You'll probably get better machine learning descriptions from other people than from me, but this is just a very broad overview that I had to put in because I am talking about some of the machine learning that's used in different neuroimaging presentations. So there are a lot of them and more all of the time, right? And there's both supervised ones where you can know more of the data that's going into the model, and unsupervised ones, which are like totally black box. It'll give you an answer, but you have no idea why, and others as well. And so when you're using it for neuroimaging, like finding out or being able to diagnose, so using the different features to say somebody does or does not have autism, if you're using one of those black box ones, you have to be very circumspect about it because you're probably just confirming what you already know in the database, right? A boy is more likely to have autism, so being male is going to make it more predictive that you are going to end up having an autism diagnosis. You have not learned anything, right? And it's not specific to the individual. But this is a new study. Don't read this study, it's actually a pretty crappy study. I don't believe anything that it actually says. But what they did, which I find interesting, is that they took, and now it's a group with ADHD, schizophrenia, and bipolar disorder, and they did a similar thing where they tried to classify them, and but what they looked at is the ones that they got wrong and trying to see why were the reasons that they got it wrong. And I find that kind of an interesting approach, right? Both looking at what kind of information is going into your machine learning and what both neuroimaging wise and otherwise in your model is causing you to misclassify people, those outliers, identifying outliers in your model. And they started proposing, why would that be? Why was ADHD often misclassified as bipolar disorder? what about those people and their brains was different compared to the overall group of people with ADHD. I find that an interesting approach, even though I believe nothing in this study. So that's, and I'm going to, because I am out of time, I'm just going to mention other new imaging areas are including imaging and genetics together and trying to understand those interactions. It has gotten to the point where I have seen some studies where they've tried to do both gene environment interactions as well as genetic contributions to some of the different imaging features that we see associated with behavior. And so that's really fascinating, but again requires lots. And if you want to correct for multiple comparisons, it just gets worse and worse between imaging and genetics. What I have an interest in is trying to use neuroimaging to target non-invasive brain stimulation. So can we use imaging, and we are, and there's new techniques and statistical methods all the time to try to determine whether it's transcranial magnetic stimulation or direct current stimulation, can we better for both groups of people but also individuals better be able to target based on their imaging where to do some of those brain stimulation or interventions. Thank you.